Okay, and different types of areas. What are they? Different types of areas, especially skin, lungs, gastrointestinal tract.
ఒకసారి నెట్ లో చూడు, ఈడ 3 ప్లేసెస్ కి వెళ్ళాలంట. సంగం ఒకటి, మెయిన్ టెంపుల్, ఇంకొకటి ఏదో.
అమ్మ శ్వేత, కొంచెం మీ మైక్ ఆఫ్ చేయండి అమ్మా.
ఒకసారి
So you will patient get any tumors anywhere. For example, skin tumors can grow any part of the body: skin, lungs, gastrointestinal tract, breast, uterus, brain, blood, etc., etc. Based on their spreading nature, okay, based on their spreading nature, a scientist classified into uh, tumors into two types. Okay, what are they? Benign tumors and malignant tumors. Benign tumors especially does not spread from one place to one place, another place. Okay, benign means this type of tumors develops but cannot or does not spread one place to another. Coming to the malignant tumor, these are the tumors spread or have a capability to move to one place to another place. Okay, spread from one place to another. These are the problem, main problem with the second type of tumors, not with the benign tumors. Okay, benign tumors are not cause any type of health issues most of the times, but malignant tumors makes nuisance and uh, disease and severe effect on the system due to the malignant tumors. Okay? And furthermore, the tumors can be divided into many types based on the nature of tissue affected. Okay, based on the spreading nature, they have classified into two types, and based on the tissue affected, exactly the what type of tissue is involving in the tumor, based upon that characteristic or that feature. uh, Tumor or tumors also different types. One is that carcinoma. Okay, in this type of uh, tumor or cancer, the tissue is that affected tissue is that epithelial cells. Okay, epithelial cells. If the tumors are developed in the epithelial cells, we uh, carcinoma. Okay, if the cancer or tumors are developed in the mesenchymal or connective endothelial cells, we can call it as sarcomas, sarcoma type of tumors or cancer. And if the cancer is developed in the lymphoid tissue or tumor is developed in the lymphoid tissue, those type of cancers are called as lymphoma. Okay, lymphoid tissue, lymphoma. If the carcinoma or tumors developed in the WBCs, okay, that type of cancer or tumor called as leukemia. Okay, based upon the place or the tissue affected, they have categorized into different types of cancers. So tumor cells are categorized by uncontrolled growth. Okay, characterized, not characterized, characterized. The specific characteristics of tumor cells are that uncontrolled growth is the one of the specific character of the tumor, and rapid multiplication, okay, is another characteristic of the tumor cell. By these, by seeing these characters, we can say these cells are tumor cells. So what are they? First one is the uncontrolled growth. Okay, these are the abnormal cells, because in general in normal cells, up to some number, up to extent, they are having the controlled growth, but this type of growth not having uncontrolled, this growth is not having uncontrolled level. Okay, and rapid multiplication. Okay, rapid multiplication means speed. Okay, innumerable cells will be generated after starting the mitotic division. Okay, uncontrolled growth and rapid multiplication. And invasiveness means it moves and it migrate. Okay, formed tissue or cells can having the ability to move one place or migrate to one place to another. Okay, this is invasiveness. It can invade any tissue from one place to another place. That is the invasiveness. That is the main. These are the three types of main characteristics of the tumor cells. Okay. They behave like embryonic cells. This is also very very important. They behave like embryonic cells as they have originated from differentiated cells by de-differentiation. They behave like embryonic cell. What embryonic cell do? The embryonic cell rapid growth will be there in the embryonic cell, and rapid multiplication can also be there in the embryonic cell. But invasiveness is not there in the embryonic cell. Like that, these tumor cells also having the embryonic cell nature, which are the cells undergoes de-differentiation. Okay, what is de-differentiation? Differentiation means some cells are not having mm, uh, proliferation uh, property after getting one stage. We can say differentiated cell. Sometimes what happens that in this type of tumor cells, in general, they are not having different any proliferated property. Okay, dividing property. Already differentiated cell. Okay, they have developed from embryonic cells, fully differentiated stage in general. But what happens in case of tumor cells, this differentiation property is disturbed or cancelled, okay, destroyed. So they undergoes the differentiation means they attain uh, uh, replication property. They attain replication property or they retain their mitotic division property. Okay, once the cell convert into the tumor cell, that is the specific case. They are abnormal in their function and have lost their intracellular contract. Very very important point. Intracellular uh, contact, not contract. Contact is not there in the type of tumor cells. Okay, what is intracellular contact? Okay, for example, in the whenever this normal cell undergoes the proliferation or mitotic division, and this cell so when the tumor cell in general, what happens that in general the normal cell under whenever this cell undergoes the mitotic division, in this division in generally the dividing cell, when the dividing cell go and touches with the adjacent cell, it stops its division. That is the main characteristic feature, that is contact inhibition. We can say contact inhibition. In general, whenever the two surfaces or two adjacent plasma membrane touches to each other, the mitotic division will be stopped after touching to each other. Okay, two adjacent cells. But if this property is lost in the in case of tumor cells. In case of tumor cell division, this type of property is lost due to the contact inhibition. Uncontrolled or rapid multiplication or uncontrolled growth is there in the tumor cells because of the contact inhibition, loss of contact inhibition. This is very very important point. Loss of contact inhibition. Loss of contact inhibition is a major cause for the uh, tumor cell conversion. There are many types of causes are there, and variety factors, variety types of factors also be there to converting the normal cell into the tumor cell. So the common mechanisms of carcinogenesis involves the transformation of proto-oncogenes. Okay, all the persons are having proto-oncogenes. Okay, any living person uh, is having this type of oncogenes. Okay, we can call it as proto or inactivated oncogenes. Okay, and these proto-oncogenes, whenever the proto-oncogenes convert into the oncogenes, the person gets cancer. Okay, the person gets cancer, or a tumor cells will be formed. And what are the causes to conversion of the proto-oncogenes into the oncogenes? There are many causes are there. Spontaneous induction means mutation. Okay, spontaneous mutations is the one of the cause to convert the proto-oncogenes into oncogenes. And another main reason is that a conversion, okay, induction, not a conversion, induction by viruses. In the induction of virus by viruses, uh, different types of viruses are very play a key role in the conversion of the proto-oncogenes into uh, oncogenes. Okay, whenever these uh, viruses enters into the cell which having the proto-oncogenes are converted into, in the presence of these viral particles or viral protein molecules, they converted into oncogenes from proto-oncogenes. Okay, for example, like adenoviruses, most of the adenoviruses are main uh, causing agents for the conversion of proto-oncogenes into oncogenes. Okay, for example, in this image, we will have some examples for the adenoviruses or oncogenic viruses. Epstein-Barr virus, also known as human Epstein hyper herpes virus. Okay, human herpes virus, we can also call as human herpes virus, and genomic is double-stranded DNA herpes virus. Most of the Burkitt's lymphoma and nasopharyngeal carcinomas are caused due to the infection of the double-stranded DNA herpes virus. And most of the hepatocellular carcinomas are formed due to the infection of hepatitis B virus. Hepatitis B virus, the main causative virus for the hepatocellular carcinoma. And most of the adult T cell leukemia is caused due to the infection of the human lymphotrophic viruses. Okay, and positive standard, single standard RNA retroviruses are main causing agent for the adult T cell leukemia. Most of the cervical cancers and penile cancers, some other anogenital cancer, neck cancer, head cancers are formed due to the double-stranded DNA papilloma viruses. So whenever these viruses goes and contact with the cell, and cell surface uh, antigens, okay, normal antigens rashes out and vanish out from the cell surface, and the special new receptors, new molecules of the receptors are formed on the surface of the normal cell and convert into the carcinoma or tumor cell. Okay, these are the very dangerous viruses, and some hepatocellular carcinomas, some lymphomas are formed due to the hepatitis virus, C virus. Sorry, and Kaposi sarcoma, primary effusion lymphoma, some multi-centric caslemia diseases are formed due to the double-stranded DNA herpes virus. Most Merkel cells carcinomas form double-stranded DNA polymorph. I am just talking about the examples. Don't bother about the names and diseases of the viruses. Just remember that viruses causes. Okay, viruses converts proto-oncogenes. Viral infection may convert the proto-oncogenes into the oncogenes. Okay, which is very very dangerous for our health. And sometimes another cause is that chemicals. Okay, chemical agents also uh, play a key role to make into the uh, to convert into the normal uh, proto-oncogenes into the oncogenes. Some of the chemicals are there. Okay, what are they? Acetyl dehyde, acetaldehyde, and aflatoxin, arsenic. Okay, you know about aflatoxin, arsenic. Okay, and asbestos, benzene, benzopyrene, 1,3 butadiene, cadmium, chromium, coal tar, dioxins, estrogens, okay, ethanol, Helicobacter pylori, releasing chemicals, ionizing radiation, formaldehyde, radon, vinyl chloride. These are the some chemicals recognized by the IARC as a uh, oncochemicals, okay, carcinogens, okay, human chemical carcinogens. They may or they can induce the uh, conversion of the proto-oncogenes into oncogenes. These are the many major causes for the turning into the proto-oncogenes to the into the oncogenes. Okay, so tumors antigens. Okay, we are been talking about tumor antigens. Okay, this is a major part of in the tumor immunology. Okay, what are the tumor antigens exactly? Okay, in the human being or any other person's tumor cells become completely altered. Okay, we know about the uh, alteration. Okay, we know the definition of this tumor. These are the completely altered from normal cells and behave entirely different from normal cells. Okay, abnormal cells. Okay. We can also tell the tumor cells are abnormal cells. During the tumor development, cell losses. A very very important point is that tumor in the during the tumor development, what exactly happened? A cell losses all its original antigens. Okay, cell losses all its original antigens, and a completely new set of antigens are expressed on the surface of this cell. What happens exactly? During the tumor development, a cell loses its natural or original antigens. Okay, on the surface of the any cell, you can any nucleated cell, different types of surface molecules and different types of surface receptors are there on the cell natural cell, and these cells in these cells, what happens in general, whenever the cell getting into the uh, abnormal, the original or its original or natural cell receptors or cell antigens, we can say cell antigens in general, we can say all the cells are having surface molecules. Those molecules are called as cell surface antigens. So what happens? New set of antigens, new set of antigens are expressed due to the above mentioned causes. Okay, what are they? Due to the infection of the virus, due to the irradiation, due to the chemical treatment, different types of causes are there, and due to the mutation, okay, these surface antigens are replaced entirely with the different molecules. These antigens expressed by tumor cells are called as tumor antigens. In general, two types of tumor antigens are there and been identified on tumor cells. Two types of antigens are being identified, and they are tumor specific antigens. One is the tumor specific antigen, TSA. What is that? Tumor specific antigen. One type of antigen. And another type of antigen is that tumor associated antigens. Okay, TAA, tumor specific, tumor associated. What is tumor specific? Tumor specific antigens are unique to tumor cells. These are the unique to tumor cells. I will just uh, show you one image to reference for the reference. The tumor, what is tumor specific and what is tumor associated antigen? Okay, tumor specific antigens are unique to tumor cells. Okay, these type of the antigens are entirely unique for the tumor cells and do not occur any surface of the normal cell, okay, in the body. These antigens may have originated by mutation, especially these antigens originated by the mutation process. And coming to the tumor associated antigen, okay, are not unique to tumor cell. Okay, these are not unique to tumor cell. And based on the origin and nature, these are divided into the two types, okay, especially three types. They have given three types in your book. And viral or chemically induced antigens or tumor associated antigens, and oncofetal antigens also comes under tumor associated antigens. Differentiation antigens, okay, differentiation antigens also comes under this tumor associated antigens. You can see the difference, exact difference between these two antigens in the image. Coming to the tumor specific, expressed by tumor cells, all the tumor cells express this tumor specific cell antigen, not present on in normal host cell. This tumor specific antigens not present on the normal cells. Okay, unique to the tumor cell. Arises mostly from oncogenic driver mutation that generate novel peptide sequences. Okay, a neoantigens. This main reason for the uh, induction or conversion of the normal cell into the tumor cell is that mutation. Main reason is the mutation. Can also be generated by oncoviruses. Sometimes, not always. Most of the times, mutations are the main reason, and sometimes can also be generated by oncoviruses. For example, alpha fetoprotein. Okay, this is the fetal gene expression in the germ cells, tumor, and hepatocellular carcinoma. In case of these two types of antigens, uh, these are the two types of antigens comes under tumor specific antigens. You cannot see these type of antigens in normal cells. Coming to the tumor associated antigens, these are the self antigens expressed by tumor cells present in the subset of normal host cells. These type of tumor associated antigens also be there in the other host cells, not like tumor specific antigens. Arise mostly from genetic amplification or post translational modification. In this amplification, genetic amplification and post translational modifications, some errors may occur and it convert into those antigens converted into the tumor associated antigen. Tendency for expression that he is higher and preferential for tumor cells. Okay, most of the times these tumor associated antigens uh, most of the time tendency for expression and that is higher and preferential for tumors. For example, melanoma associated antigens, okay, and especially in the testes along with the malignant melanoma. And these are the few examples for the uh, tumor specific and tumor associated. According to your book, tumor specific antigens are unique to the tumor cells and do not occur on the normal cells in the body. They these may be induced by viruses sometimes or carcinogens. Okay, for example, they have given one example. What is that? Uh, Viral or chemical induced tumor specific antigen conversion. When a tumor is induced by the virus, the antigens of the infecting virus appears on the surface of the tumor cell. These antigens are called viral specific antigens. Okay. In addition, new antigens appear on the surface of the viral infected tumor cells also. These antigens are called tumor specific. Two types of antigens are there on the infected cell surface. Okay. For example, one is that viral specific antigen. Okay. These are the viral specific antigens. One of the uh, specific antigens are there, and other antigens also be produced by the infection of the virus. And what are they? We can also call as tumor specific antigens. Two types of antigens are there. You can see in this picture tumor associated, tumor specific. In the tumor specific, two types of antigens are there. Okay, this is one and this. And so oncoviral antigen, private neoantigens. Okay, oncoviral antigens are there. Private neoantigens are there. The two types of antigens are there in the tumor specific antigen along with the viral antigen. For example, uh, RNA viruses, tumor antigens are there. Human T cell lymphotrophic viruses. Coming to the DNA virus, okay, Epstein-Barr virus is the best example for tumor or chemical-induced antigen, virus-induced antigen. Human papilloma virus, hepatitis B virus is the best example for the virally induced tumor antigens. These are the different types of antigens. Antigens may lead to the different types of carcinomas in the human being. And coming to the tumor, cells induced by the different chemicals expresses a new type of antigen called tumor specific transplantation antigen. Okay, these antigens are expressed due to the mutagenesis of the DNA by certain chemicals. I have shown uh, the different types of chemicals in the previous slide. Those chemicals are responsible for the conversion of the different types of the DNA mutations. Okay, normal sequence is undergoes the different types of mutation, and this may lead to the uh, normal cell conversion into the abnormal cells. So that is tumor. Okay, and in general, what happens at surface? Normal cell with self peptide on class one molecule, MSC molecule. Uh, Coming to the other molecules, altered cell peptide expression of the mutated gene into the new peptides, okay, is the one of the reason, and expression of viral protein also one of the another reason for the conversion, and oncofetal inappropriate expression of embryonic gene. Okay, inappropriate expression of embryonic gene also may leads to the conversion of the surface antigens. So inappropriate expression means normal cell surface molecule production uh, is disturbed due to the different types of reasons, and inappropriate protein expressed on the surface of the cell, and that is especially embryonic cell. So that may lead to the fetal oncogenes, okay? Production of fetal oncogenes. So this is about tumor specific antigens. Coming to the tumor associated antigens which are not unique to tumor cells, what we have discussed earlier in the table, may be proteins that are expressed on the normal cells during fetal development. Okay. During the fetal development, this type of the tumor associated antigens will be seen during fetal development. And when the immune system is immature, okay, when our immune system is immature, especially in the fetus embryonic stage, in the fetus, this antigens unable to respond. Uh, this our immune system unable to respond, but that normally are not expressed in the adults. Okay, in general they are not expressed in the adult, but adult stage, but they are already expressed in the fetus. Okay, when our immune system not get activated, so exactly not get formed correctly. So reactivation already they are formed, They have been formed in the fetus stage, okay, embryonic stage. Reactivation of these embryonic genes that encodes the proteins in the tumor cells result in their expression on the fully differentiated tumor cells. Okay, sometimes these embryonic tumor cells, uh, embryonic genes, okay, gene products, okay, gene cells suddenly reactivated and undergoes the differentiation and produces its products on the surface of the cell. Okay, different types of antigens are produced on the surface cell. They also acts as a tumor associated antigen. Okay, they may be proteins that are normally expressed in the extremely low level on normal cells, but are expressed at much higher level on tumor cells. Generally, with compared to the normal cells, the tumor antigens are more in the tumor cells. Okay, what are oncofetal antigens? Oncofetal antigens are not unique to tumor cells. Okay, these are the antigens are not unique for the tumor cells, since they are also found on the cells during embryonic. Okay, we are having this type of genes or cells in our body. Okay, but they are not expressed or they not reactivated. Okay, so these antigens are there in our in healthy persons also, but they not get activated. So we cannot call as tumor specific. We can call as tumor associated. And since they are also found on the cells during embryonic development and are found at a very low level means our immune system cannot recognize us, or these low level cannot elicit the immune response in general. Therefore, these are called as tumor associated antigens. And these carcinoembryonic antigens are one of the example for the tumor and oncofetal antigen, CEA, carcinoembryonic antigen. Every persons are having this type of antigens. This antigen is expressed by many gastrointestinal derived tumors. Some tumors are developed or expressed after some period of time, including colon carcinoma, colon cancer, and pancreatic cancer, liver cancer, and gallbladder tumors are having this type of antigens, especially. All these areas are having this type of oncofetal antigen inactivated. Okay, are sufficient insufficient number of or low level of antigens are there, are spread in this area. Whenever this uh, area is get activated, cells are get activated, this number of cells are enhancement of the growth, and rapid multiplication will be there after some causes. Okay, after getting signal from the different types of causes, and these rapid multiplication and uncontrolled growth will be there due to the presence of previous present of the carcinoma embryonic antigens. Okay, all these carcin areas are contained having these type of carcino embryonic antigens. Where exactly they are there? Gastrointestinal area, including colon, okay, pancreatic region, liver, gallbladder areas are having this type of uh, carcinoembryonic antigens. Whenever the different types of uh, causative agents like viruses and different types of irradiation and different types of chemical induction may converts these carcinoembryonic level, a very low level to an sufficient level to uh, conversion of the proto-oncogenes into the oncogene. This is very, very important. And it is also expressed by the gut, liver, and pancreas of the human fetuses. The human fetuses are also having this type of proto-oncogenes. Okay, we can call it as proto-oncogenes, carcinoembryonic antigens, and alpha fetoprotein, AFP, also there in our body. in all the human beings. It is found in the secretions of yolk sac. Okay, at our fetal stage, these are all already developed. All these type of antigens already developed, and fetal liver epithelium as well as in the serum of patients with liver tumors. They have found uh, whenever these type of patients have examined with the different types of techniques, they have found this type of antigens. Okay, special antigens like oncofetal antigens (EEA). Don't bother about full forms, abbreviations of these short forms. Just for your reference, I have given specific tumor markers: CEA antigen, TPA antigen, CSLX antigen, CA15 P 3, PSA, SAN1, etc., etc. These are the oncofetal antigens. They are there in our body at the stage of fetus, embryonic stage, at the stage of embryo. So, at the stage of embryonic stage, all these antigens are developed on the cells. Okay, antigen cells are developed. So, different types of oncoplacental antigens are there. Okay, cancer vessels also there. These are acting as a antigens. Okay, a way, in the future they convert into the proto-oncogenes into the uh, oncogenes, normal to abnormal, due to the different types of causes. Since these antigens are not specific to tumor cells, they can be elevated. Okay, they can be elevated in non-neoplastic diseases such as chronic inflammation of the bowel, cirrhosis of the liver. Okay, if it is there, some persons are suffering from the chronic inflammations like bowel inflammation and cirrhosis inflammation. Since they are not converted into the tumor cells, but they some persons, okay, some persons, those are not having the capacity to fight with the this type of antigens, may get the inflammation at the bowel, in the bowel area, and cirrhosis of the liver also can be seen this in this type of patients person. For example, uh, you in your book they have given two types of antigen, that is carcinoembryonic antigen (CEA) and alpha fetoprotein AFP. These are the antigens are identified in the gastrointestinal uh, tumors. Okay, most of the gastrointestinal tumors are having this type of antigens, and in the breast cancer also, they have found this type of antigens. Finally, conclusion is that presence of carcinoembryonic antigens may lead to the gastrointestinal cancer or breast cancer. Okay, and alpha fetoprotein ATP. Okay, uh, these are the glycoprotein in nature and may lead to the hepatomas. Most of the hepatomas cancer are having this type of alpha fetoprotein in major part. Okay, uh, coming to the immune response, these are the antigens. Different types of antigens are there. We have uh, studied about the tumor specific antigens, tumor associated antigens. These are the two types of antigens that we know. So, what is the response, our immune response against the tumor? Okay, what type of immune response uh, can be taken by the our immune cells over the tumors, over the tumor or uncontrolled cells? Okay, every person, remember, every person are having the tumor antigens, but fighting against tumor cells is very, very important. Immune response. Okay, if you have enough immunity to fight against the tumors, you may not get cancer. If you not having uh, enough immunity against the tumor cells, the person get cancer. Okay, that is the immune response play a key role to suppress the tumor cells, so to suppress the formation of tumor cells or cancer, most of the cancer. So tumor cells are completely altered from the normal cells, we know, and they express new set of antigens. Okay, because we are calling the tumor cells because they set total entire design of the plasma membrane antigens are redesigned. The antigens are replaced entirely. Hence, a tumor behaves as an allograft. Okay, for our immune system, this tumor cell is acts as a allograft in general. Okay, what is allograft? Allograft is a graft in which uh, own body part is replaced. Donor is one. Donor from the donor to recipient. Donor is only one person, and recipient also same person. From in the same person, body a tissue from one area to transplantation of the tissue or cell or organ from the one place to another place is called as allograft. Okay. Grafting the self tissue in the person is called as allograft. So we know about the allograft rejection and the action mechanism of the action of immune response same as allograft rejection. This is the meaning of the allograft, the comparison between the allograft and tumors. Our immune system recognizes as allograft. All the tumors are recognized as a allograft by our immune system. So fighting mechanism is same as allograft rejection. The immune system recognizes is a non-self, and all the tumors are recognized as a non-self and induces immune responses. Okay? And it is having the capacity to induce the immune system because it has uh, uh, different it design is replaced by the different types of another surface molecules. So our immune system recognizes as a non-self, and also tumors surface molecules can have having the capacity to induce our immune system. So and it induces immune response to kill or remove the tumor cells. The anti-tumor immune responses develop into tumor-bearing patients. Thus, the anti-tumor antibodies and T cells, okay, anti-tumor antibodies are being developed due to the presence of the different surface antigens, different type of surface antigens. Those antigens are recognized as a target antigen, and for that, specific antibodies are produced, and specific T cells are generated, and all these may lead to the destruction of the killing of the tumor cells or tissue. And these, along with the non-specific immune defense mechanism, the innate immune branch of the innate immune branch also play a major role, along with the acquired immune branch of the immunity. And these two types of antigens, TSA, and TEA, will be processed after the conversion by our different types of antigen presenting cells. For example, macrophages, dendritic cell engulf the total cells or totally differentiated cells engulfed by the macrophage and dendritic cell present a processed antigens or TSA antigen, TAA antigen process and presented with the MSC molecule 1. Okay. In case of macrophage, in case of dendritic cells, MHC2. Okay. This is the difference between professional antigen presenting cell and normal antigen presenting. In case of our normal body cell, nucleated cell, these TSA and TAA presented with the help of MHC molecule 1. In case of other cells, MHC molecule 2 will take care about the presence of antigens, so TSA antigen, TAA antigen. So after presence of the antigens with the help of MHC molecule, what happens? Different mechanisms. Okay, different mechanisms are get activated. So target tissue or target cell will be damaged or killed, necrotized, by the different mechanisms. What are they? What are the mechanisms are involved in the different types of killing processes? For example, by antibody complement system. We know about all these mechanisms. No, any mechanisms are not new to our study. So all these are mechanisms. Are you well know about these all mechanisms? We will discuss one by one. Page number 218. See the page number 218. By antibody and complement system. Okay? The tumor antigen, okay, tumor cells shading antigen. Okay, tumor cell will release antigen or express the antigen, and B cell binds to tumor antigen. Okay. B cell having the has a capability to recognize the antigens on the surface of the different types of cells. So B cell go and binds with the tumor antigen, and this binding facilitates the activation, like sensitization and activation and proliferation of the B cell, and a specific tumor specific antibodies are produced after activation of the B cell. Tumor specific antigens antibodies are produced. These antibodies go and binds with the tumor remaining tumor cell surface antigens. So in this case, tumor binding of the antibodies and with the tumor antigens, tumor antibody antigen complexes are formed. We know what happens after complex formed, antigen antibody complex formed. What happens? Wherever the antigen antibody complexes are there, the complement protein recognizes these antigen antibody complexes, okay, and goes and binds with the FC region receptor of the complement protein. And what happens after the attachment to the FC region or complex with the complement protein? The cascadial reactions are get activated, leads to the membrane attack complex formation on the surface of tumor cell. Okay, cascadial reactions after few cascadial reactions, it forms like our complement proteins forms one pores or different pores areas on surface of the plasma membrane, different regions. Membrane attack complexes are formed due to the pore formation. The cytoplasm will be oozes out from the cell. Is so naturally that those type of cell undergoes the death. Death will be or killing with the killing due to the formation of the pore on the surface of tumor cell. This is one way to kill the tumor cell. Understand, Amma? This is one of the
Yes, sir.
mechanism to kill the tumor cell. And we have our body or our immune system also having the different types of mechanism to eliminate or kill the tumor cells. For example, antibody dependent cell mediated cytotoxicity. Okay, we also know about we also studied this type of antibody dependent cell cytotoxicity in the tumor hypersensitivity and graft, that means transplantation. In these two topics we have clearly discussed about this ADCC. Okay, once again I am going to repeat about ADCC. What is ADCC? Antibody. Okay, antibody uh, play a key role to finger the uh, tumor cells. These all tumor antigens fingered out by the with the help of antibody. Wherever the tumor antigens are there, this antibody goes and binds with the antigens. This is help as a fingering or targeting molecule. Okay, this is the target area. Okay, help to recognize the target for different types of cells. What are they? One side, for example, FAB, FAB region go and attaches with the tumor antigen, and this attachment attracts the different types of cells. For example, neutrophils, okay, eosinophils, basophils, macrophages, natural killer cells are having FC receptors on its surface. If those cells are get attracted towards the binding of the FC region, okay, binding of the antibody to the tumor antigen, and all those cells comes and binds with the FC region of the antibody and get targeted. The tumor cell get targeted by the different types of active immune cells. For example, if the FC region, if the macrophage FC region goes and attaches with the FC region of the antibody, what is the consequence? Tumor necrosis factor, a most dangerous chemical, tumor necrosis factor, will be released from the macrophages. It can also kill the tumor cell directly. If the natural killer cell go and bind with the FC region of the uh, tumor anti attached antibody, what happens? This I will show an image for you. Different types of cells releases some different types of molecules. Okay, FC region, okay, FC receptor having cells releases encounters on the tumor cell due to the attachment of the FC region to the surface of the uh, inactive cell. Whenever this FC region attaches with the cell, this attached cell get activated and reacts upon the tumor cell. Okay, where exactly antibody attached with the FC region, FAB region, with the help of FAB region. So macrophages kills the uh, cancer cell by releasing tumor necrosis factor, and natural killer cell also releases the different types of porphyrins. Okay, also kills the cell by forming the pores on the tumor cells, and CD8 cells. Okay, not in case of this antibody dependent cell type toxicity, and different types of neutrophils, basophils, and eosinophils are having FC receptors. All these cells releases its active ingredients on the surface of tumor cell, and tumor cell will get damaged. Okay, so this is one type of antibody fingers the target, and remaining cells kills the target. This is the main theme behind the ADCC. And what is another type of mechanism? By cytotoxic T cell. This is very most important uh, mechanism to kill the tumors formation, tumor tissues or areas. A specific cytotoxic T cell recognition of a TAA or TSA after process and presentation by the MSC molecule 1. MSC molecule 1 present the TAA and TSA antigens to the TC cells. What are the TC cells? T cytotoxic cells, which are having T CD8, CD28, and CD8 molecule on its surface. The binding of these MSC and CD8 and CD28 receptor, and B7 with the CD28 receptor. These receptors cross binding activates the T T cytotoxic cell. What exactly happening? Cross binding of or binding of the B MSC one molecule with the CD8 marker, CD8 receptor, and gives the one signal, and another signal, a co-stimulatory signal for the activation of TC cell is that B7 surface molecule on the antigen presenting cell to the CD8 CD28 receptor on the surface of TC cell. These TC cell receptors and antigen presenting receptors communicate with each other, gives as a stimulatory and co-stimulatory signals to activate the TC cell. Finally, TC cell get activated and releases different types of hazardous enzymes on the surface of tumor cells. What are they? Porphyrins makes pores on the surface of the tumor cell, and granzymes What granzymes do? Granzymes go and binds with the mitochondrial enzyme, that is caspases, and inactive caspases get activated and may lead to the natural death of the tumor cell. Okay. This is how the cytotoxic T cell play a major role in the tumor killing of the tumors in our body cells. Okay. So this is one of the mechanisms. Any doubts, Amma?
No, sir.
No, sir.
If you have no doubts, I will move forward. So we have another mechanism is that by activated macrophages. I have told you earlier, different mechanisms are uh, uh, entirely patrols. Okay, patrols for the tumor and presence of the tumor antigen, and not only patrols but also kills the tumor cells by different mechanisms, activates the different mechanisms through the different mechanisms. So, by activated macrophages, also one of the major mechanism to kill the tumor cells. What happens in this case of macrophages activation? Tumor cells shedding antigens means tumor cell wherever the tumor cell antigen, tumor antigens, for example, TAA presence and TSA presence. If the cell surface present with the if the TAA antigen, TSA antigens are there on the surface of the tumor cell, these macrophage go and bind with the surface uh, molecule, the surface antigen, and engulfs the entire cell. Okay, engulfs the entire cell. That is one of the mechanism we know. And what happens in case of in another case, and whenever these antigens binds with the macrophage, and these macrophage, macrophage naturally engulfs. Okay, that is the only way to present the antigens to the TH cell. It presents it antigens. Okay, TAA antigen, TSA antigen presented by these macrophages to TH cell. Okay, we know this is a cell mediated immunity. Okay, cell mediated immunity branch comes under cell mediated immunity branch. Whenever this macrophage present the antigens to the TH cell, and these both cells get activated by taking the signal from other cells. Okay, for example, TH cell releases the interferon gamma. The TH cell releases the interferon gamma, and these interferon macrophages are having the interferon gamma receptors on its surface. It receives the interferon gamma molecules, and whenever these interferon gamma molecule received by the inactivated macrophages, these macrophages get activated completely and releases tumor necrosis factors. A tumor necrosis factor on the surface of tumor cells, not CS cell, on the surface of tumor cells. Okay, this tumor necrosis factor will be released after getting activated by the interferon gamma, which is released by the TH cell, and this tumor necrosis factor kills the tumor cell, okay, by different ways. This is one mechanism. The activation of the macrophage may lead to the killing of the tumor cell. Okay, and we have another mechanism that by stimulation of natural killer cells. Natural killer cells also play a key role in the killing of tumor cells and entirely. The tumor antigens, TA antigen, TSA antigen can be recognized by directly by the natural killer cell, and these natural killer cells go and bind with the antigen, and the binding precipitates the activation of the natural killer cell, and these may lead to the aggressive ingredients released on the surface of the tumor cell. Tumor cell will undergoes the lysis, and this is the main major mechanism by the natural killer cell. Okay, attachment of tumor cell with the natural killer cell may lead to the aggressive chemical mediators, okay, pharmacologically active ingredients released on the surface of the tumor cells, and tumor cell undergoes the lysis. These are the mechanisms. Different types of mechanisms are having play a key role in the elimination of the tumors. So what happens in case of cancer patients? Okay, we have excellent mechanisms to protect from tumor formation in our body. But some persons, some extent, getting the cancer or getting the tumor because our immune response, our all these antigens, the tumor antigens elicits the immune response against them. Their exit, okay, their exit several evasion mechanisms. Okay, tumor antigens are tumor cells develops the escaping mechanisms from our immune system. They develop escaping mechanisms. This immune response can only happen when the antigens are expressed or antigens are recognized by immune system or immune cells. It happens. It can only happen after recognition. But what happens sometimes, these tumor cells are developed uh, the different mechanisms to to escape from the to evasion from the our immune system. Sometimes they escape. That may leads to the cancer. Immunologic enhancement of tumor growth. Okay, this is one of the escaping mechanism. Modulation of tumor antigen is sometimes some part of the antigen may be modulated after getting exposed with the immune system. That at that time our immune system may not recognize as the antigens of the tumor cell. Reduction of class 1 MSC molecule on the surface of the tumor cells. Okay, sometimes it is one of the way to escaping mechanism for the from the our immune system. Lack of co-stimulatory signal. All the cells, immune cells, requires co-stimulatory signal to activation process for the total activation. If you take B cell, if you take T cell, take macrophages and different immunological cells, wants or requires co-stimulatory signal, then only they get activated and total activation process will be completed. Then and the cells go undergoes the proliferation. So lack of co-stimulator, these cells will not give the co-stimulatory signal to the immunological cells. Okay. Whenever these tumor cell go and bind with the macrophage or B cell, it cannot give the co-stimulatory signal to activation of the for activation of the other cell. No co-stimulator signal, there is no activation of the other active immunological cell. No activation, no immune response. So that is the way, one of the way to escaping from the immune system. If it is escaped, may leads to the tumor formation. Okay. And in spite of this, it is clear that an immune response can be generated to tumor cells. Therapeutic approaches. Okay, we can recognize this type of misleading, okay, escaping mechanisms. Though they are escaping from the our immune system, we can recognize those type of illnesses, okay, developments of tumor developments in the therapeutic approaches by using different therapeutic approaches. And what is tumor? By this tumor surveillance, we can recognize those type of escaped uh, formed tumors by therapeutic approaches by uh, immune surveillance. This is what is immune surveillance? Immune surveillance is the process by which the immune system monitors. The immune system monitors and destroys cells, tissues, and foreign substances that are abnormal or harmful to the body. This is what is going on 20 by 24 by 7 in our body. Immune surveillance is going on 24 by 7 in our body. What is immune surveillance? The careful watching. What exactly the meaning of surveillance of immune system? The careful watching, identification, killing, These are the three steps, major three steps. Entire these are collectively called as immune surveillance. Careful watching of foreign invaders or foreign substances, and a careful identification of foreign non-self materials, the careful killing of other like tumor cells by immune system is called as immune surveillance or immunological surveillance. It is a word proposed by the Paul Eldridge, okay, nearly in the early 1900 year, 1900, 19th century. He suggested that immune system recognizes cancer cells and eliminates them. First time, he suggested that immune system play a key role in the recognition of the different types of tumors. And another eminent personality, Louis Thomas, suggested that cell-mediated branch of immunity uh, patrols, patrols the body, entire body, for the and eliminate the cancer cells. These two persons, uh, extensively worked on the cancer tumor immunology. When the immune system fails to recognize and kill the tumor cell, the tumor cell succeeded and multiply and develop into cancerous growth. If a cancer develops into it is because of abnormal cell has escaped. Okay, escaping of abnormal cell may leads to the from there it escape from our immune system, from the immune surveillance. Escaping of abnormal cell from the immune surveillance may leads to the cancer. Okay, exact meaning is that escaping of abnormal cell from the immune surveillance may lead to the cancer development. The escape of the tumor cells from the immune surveillance may be due to the rapid multiplication. One is major cause is that rapid multiplication and growth of the tumor cells may lead to the escaping. A tumor antigen may coat the lymphocyte. As a result of lymphocytes cannot act on the tumor cell. Sometimes it may antigens may coat on the surface of lymphocytes. So lymphocytes cannot having the recognition. It loses its property to recognize the another antigen. So escaping of different types of abnormal cells may lead to the from the our immune surveillance may lead to the development of different types of cancer. And how we can recognize these cells? We can recognize these type of escaped cells. Those are formed as a tumors by the immunodiagnosis. Okay, by the immunotherapy. Okay, we can diagnose these type of tissues or tumors by the different types of processes, immunodiagnosis. And in this process, what they do, and they construct the monoclonal antibodies. They synthesize the monoclonal antibodies to the tumor antigens. Okay, these tumor antigens for the surface antigens, for the surface monoclonal, for the surface tumor antigens, these monoclonal antibodies uh, will be developed under the laboratory condition, and uh, by this monoclonal antibody attachment, they recognizes the uh, tumors after using different types of techniques. Okay? After the imaging, after tumor imaging, after searching for the different types of monoclonal antibodies, these monoclonal antibodies goes specific to the different types of uh, tumor antigens, and they go and bind with the antigens, and they influorinated. Okay, after fluorination, fluorescent compounds addition, these monoclonal bodies were attached with the different types of fluorescent molecules, and the in the imaging they get where exactly the monoclonal antibodies uh, are there. The presence of monoclonal antibodies, uh, they in the imaging, they guess the where exactly these antigens are there, where exactly these tumor cells are uh, developing. Okay. Presence of aggregation of the monoclonal antibody in the imaging. Okay. In the fluorescent image, they recognizes the areas where exactly tumor cells are developing. That is the diagnosis part. Okay. By monoclonal antibodies, and by the different types of fetus antigens, tumor oncofetus antigens will be recognized with the help of this monoclonal antibodies, and different types of markered uh, monoclonal antibodies are radioisotopes linking with these monoclonal antibodies with the radioisotopes like iodine 131 monoclonal antibody specific for the appropriate TAA antigen intravenously injecting, and these after some time the images will be taken up from the patient, and in the image tumor metastasis using this image will be identified wherever these monoclonal antibodies are aggregated, and those areas may in those areas the specific type of the tumor is developing. The result is that from the monoclonal antibodies they recognizes the different types of cancer area. So, and immunotherapy, what are the immunotherapies are there to protect the body from the cancer? Different types of immunotherapies are there, supplement these natural defense mechanism. Okay, under the development of different discussed mechanisms. Immunotherapy of tumors using transfected tumor cells. Transfected tumor cells can be used as a one of the therapy to uh, protect the tumor patients, cancers from the different types of the patients from the different types of the cancer. And another therapy is that using adjuvant, weakened and uh, our immune surveillance system fails whenever our immune system cannot recognizes the tumor cells, the antigen. After adding adjuvants to our immune system, there are these antigen, these uh, our body recognizes, our immune system recognizes fastly than the normal situation. After adding adjuvants, these adjuvants goes and binds with the different types of cells, especially uh, antigen presenting cells. Okay, the presentation of the antigen cells, recognition of the antigens of the antigen presenting after the addition of the adjuvant. Okay, after the addition of the adjuvant, this property of recognition, identification of the antigen uh, will be uh, will be the more than the normal time. So, addition of the adjuvants can also be one of the best immunotherapy for the uh, uh, tumor elimination, okay? Tumor recognition and elimination. And immunotherapy for tumors using recombinant cytokines, different types of cytokines, giving different types of cytokines to the for the patient is also the best option for the uh, treating the tumors. Okay, they are including interferon alpha, beta, gamma, interferon 1, interleukin 1, 2, 3, 1, 2, 4, 5, and interleukin 12, and GM, CSF factor, and tumor necrosis factor. Giving these cytokines to the patient, and expression of MSC1 is enhanced after giving these uh, cytokines to the patient, and different types of immune mechanisms also enhanced, processes also enhanced after giving these type of cytokines. Okay? Activation of the TH cell and TC cell also be taking place after giving these cytokines to patient. Okay? So all these cells get activated and recognized as a tumor cell and eliminate by the action of the different types of processes. So this is one of the therapy. And another therapy, coming to the another therapy, tumor using LAK and TIR. Okay, LA, what is LAK? And lymphokine activated killer cells. Okay, LAK means lymphokine. Giving interleukin 2. Okay, giving interleukin 2 enhances the proliferation of the B cell. Okay, artificial giving of the interleukin 2 to the tumor having patients enhances the B cell recognition of the tumor antigen identification. And after this process, lymphokine lymphocytes, especially B lymphocytes, get activated more and more efficient recognition and elimination will be taking place. This is one of the types. And tumor containing lymphocytes that have infiltrated the tumor, they can be obtained and expanded into the in vitro with the interleukin 2. These activated tumor infiltrating lymphocytes are called tumor infiltrating lymphocytes. Tumor infiltrating lymphocytes, TILS. The expanded tumor infiltrating lymphocytes can be re-injected, re-injected into tumor patients. Okay, this type of tumor infiltrated lymphocytes can be injected into the patients together with interleukin 2 that could show tumor cell destruction. After giving this TIL and TLAK along with it interleukin 2 may destruct the tumor cell. This is one of the therapy. And immunotherapy of tumors using antibodies. Also, monoclonal antibodies have been used in the various ways in the cancer. Uh, I will post if you want if you want to, I have one video lecture, video demonstration on the monoclonal antibody usage of monoclonal antibodies in tumor destruction. I will send a link to you to better understand what happens in case of monoclonal antibodies. And monoclonal antibodies are specific for the specific cancer. Not all the these monoclonal antibodies are used for the all antigens. It is especially constructed with the one type of antigens. Okay, antigens proteins on the surface the cell that can cause the immune system to respond, and these monoclonal antibodies goes and binds with the surface antigen. This can cause the immune system to attack the cancer cell, and this binding of the monoclonal antibody on the surface of the uh, tumor cell may lead to the destruction of the different types of tumor cells. For example, it attracts the different types of cells. For example, T cell and different types of macrophages, different types of the mono uh, different types of natural killer cells, and neutrophils will be attracted towards the monoclonal antibody Fc region and acts upon the tumor cell. Okay, same like our natural antibody mechanism. This antibody dependent cell type cytotoxicity mechanism. So these are the therapies to protect the patient from the tumors. So immunotherapy of tumor using vaccines also taking place. Uh, so these are the monoclonal antibodies also called as magic bullets, magic bullet therapy. Okay, in this guided missile therapy, this monoclonal antibodies goes and directly binds with the target antigens. So the designing of the monoclonal antibody like that. Okay, the guided missile. So toxic agents are delivered specifically tumor cells. Reagents known as immunotoxins have been constructed by the coupling different types of monoclonal antibodies are very play a crucial role to control the different types of carcinomas and tumors. And tumor using vaccines also is under progress. Research under progress. Numerous approaches are being used to develop vaccines for use in the treatment of cancer. The prophylactic approaches involves the use of vaccines that induce immunity to viruses, uh, shown to be associated with the development of tumors. So these vaccines are using of vaccines to treat the tumors is undergoing under under progress. Okay, this is about just basic things about tumor immunology. Just you remember the what is tumor immunology, what are the tumor antigens, okay, and what are the mechanisms, okay, what is the immune response to tumor. Just remember these things enough. What is immune surveillance and what is immunodiagnosis and immunotherapy in general. This is general topic, not depth topic. If you have any doubts, just ask me. Any doubts, Amma?
No, sir.
If you want, I will share one video how the monoclonal antibodies are being used to treat the tumor cells. I have that link. If you want that link, I will send. I have already sent to your friend. If you want, I will also send to you. Okay? Remaining number. Okay, Nam?
Okay. Okay, sir.
Okay, all the best.
Sir, what is the full form of CTL?
PTL, Amma.
Yes, sir. CTL. Precursors.
Cytotoxic lymphocyte.
Okay.
Cytotoxic lymphocyte response. Okay. Okay, Nama?
Okay, sir.
Okay. If you have any doubts, you can ask in the subsequent class, in the next week. Okay. Just read once. If you have any uh, doubts, you write down those doubts on the page. You can ask the next class also. Okay, Nama? Any
Okay,
doubt
sir.
from your syllabus? In upcoming classes, you can ask.
Okay, sir.
Okay, Nama?
Okay, sir. Thank you.
All the best.
Thank you, sir.